Hi, everyone, and thank you for joining this session today. So I'd like to first introduce the co-speaker today, Dr. Ian Durbach. Ian works with the St. Andrews University, and uh, he's the he's the motor behind this entire PAWS initiative. I'll talk a little more about what PAWS initiative is. Many of you already know about it. But Ian, would you like to please introduce yourself?
Yeah, sure. Hi, everyone. It's nice to see some familiar faces and also lots of new names for me. So I'm Ian Durbach. I'm a researcher at the Center for Research into Ecological and Environmental Modeling at the University of St. Andrews, working mainly on spatial capture-recapture methods, and with particularly for snow leopard applications. So, like Koustubh said, I've been working with the PAWS team since 2018, and really enjoy working with the snow leopard community to sort of push these spatial capture-recapture methods sort of to their limits. So yeah, today I'll be taking you through an introduction to the techniques, and yeah, looking forward to it and to interacting with you all. Thanks, Koustubh.
Thank you, Ian. And just to give you a very brief outline, what we are going to do is both Ian and I will briefly talk about, as Ian said, spatial capture-recapture methods. I'll talk about the distribution methods and a brief introduction to the PAWS initiative. The idea here is that since Justine and Raki have just given us only 2 hours in one session, we won't be able to pack everything together. So it'll be more of a skimming through several of the concepts, and we will be very happy to share through the presentations. Both Ian and I will be sharing some of the more in-depth resources that have been developed that are available online as well. But more than anything else, we would really like to have some interactions with you all. I can't promise I'll have all the answers. I can promise Ian will have all the answers, but definitely what we will try to do is we'd like to have some interaction. If you have any questions or challenges or issues that you're facing with either distribution surveys or population abundance surveys pertaining not just to snow leopards, but wildlife in general, please do join in and interact and see if we can have some interactions there. Okay, so I'm going to quickly share my screen here. Right, so let me start by saying this, and this is not an exaggeration. I find it fun to keep collecting some completely useless information once in a while. But of the last 25 people I met in the last few months, those who had never met before, 21 of them have asked me how many snow leopards are there in their country or in the world or anywhere the moment I mentioned that I work with snow leopards, or within the next 10 minutes of that conversation. Right. So in 2017, when the senior officials from the snow leopard range came together in Bishkek to decide upon the language and the details and the aspects of priorities that have to be included in the Bishkek Declaration for Snow Leopard Conservation, they expressed very serious concerns about the lack of knowledge and information regarding where snow leopards are and how many there are. After all, in fact, this is the clause, as you can see. The reason being, this information is used to make decisions and policies. And in absence of such information, which is considered to be very basic information for decision making, whether it's creating a protected area or preventing an area from being mined or built up on, wherever you go, I think the information about where the species of concern is and how many of them are there are some of the most basic and most important questions that are usually asked. So following this request from the heads of the governments of all the 12 snow leopard range countries, who basically endorsed the Bishkek Declaration, what we initiated or what we got involved in was something extremely ambitious. We call it the population assessment of the world's snow leopards. It's an ambitious initiative to estimate the wild snow leopard population across the world. It not and in doing so, we managed to bring together state-of-the-art statistical design, ecology, artificial intelligence, and the spirit of collaboration. So much so that in the last 5 years or so, the PAWS initiative has spawned a 400% increase in the spatial coverage of statistically robust and ecologically meaningful snow leopard surveys across its range. From what we knew, which was less than 2% of the entire habitat covered or sampled using replicable, reliable methods, today we have more than 10% of the entire potential distribution range covered under these surveys. But there's a very important rider here. How much you survey is really not important. And as statisticians such as Ian put it very succinctly, it does not matter how many percentage or how much area is sampled. What really matters is where it is sampled. So what really ensued along with this huge initiative was that an advisory panel was set up, which included statisticians, it included ecologists, it included people with political background, and conservation practitioners as well. So a diverse range of experts were roped in to create specific guidance, including tools and apps that people could use as per their need, be it designing camera trapping surveys or genetic sampling surveys, to guidance on how to collect genetic material or how to set up camera traps when you find a good location, and so on. So there's a wide range of tools and equipment and guidelines that were created, which are freely available on various resources. The PAWS team also developed training modules that could be delivered in person as well as online. And it's remarkable that in the last 4 or 5 years, some of these tools have not only helped snow leopard conservationists, but also people working on other species, because many of these tools and applications are species agnostic. They use methods which can be used across various species as well, as long as you can address a few assumptions. Most of you, I believe, may have already seen the online training modules and resources, but if not, please don't miss these. I will request Justine and Raki to kindly share these resources with all the participants separately so that you can freely use them. The one on the top left is an excellent, almost 160 hours, if I'm not mistaken, of a training module about using spatial capture-recapture with camera traps. And that's something that Professor David Borchers and Ian have put together. Then we do have other resources on the globalsnowleopard.org website. We have other training modules on the Snow Leopard Network website as well. So what I will do today is I'm not going to, I'm basically going to focus primarily on the distribution surveys. And as you know, species distribution and abundance play an important role in policy development, conservation planning, and also monitoring. Above all, if you have to communicate conservation, I think some of these basic maps and statistics are extremely valuable. So today, Ian and I will take you through a quick introductory tour. We won't be able to spend a lot of time, as I mentioned before, that we only have just 2 hours. And we'll need to be brief and skim through several concepts during that hour. So you may consider this session as an introduction to several other detailed training modules that have been put together painstakingly, as I mentioned earlier. So I'm going to focus on the distribution surveys, whereas focusing more so on occupancy-based surveys. And Ian, on the other hand, will be focusing on spatial capture-recapture that helps us get to the next level. Right, now distribution is typically nothing but a map which roughly or accurately defines where a species is found. Types of distribution maps, they also vary dramatically from being binary polygons, just showing presence of a species on a hand-drawn border to somewhere which could be probabilistic, showing how probable it is for a species to be distributed in. You may sometimes also come across maps that make no sense at all in the name of distribution maps, but they're there, and such maps do come up when you create a map using some statistics or GIS without really thinking ecologically or logically as well. So the distribution maps can be of a very wide range of wide spectrum of quality, value, and relevance. Historically, a lot of these distribution maps were developed based on hunting records. Sometimes these hunting records would be collected meticulously. Sometimes they would be fabricated just to show off that X and X, Mr. X and X shot a tiger in a place where it was never shot before. Sometimes they were just carried and left at a place and so on. So, but this is how historically several species distribution maps were created. Then came a stage where people started sending out questionnaires and would wait for people to report sightings of species in areas that they knew about. I remember when I'd started my PhD in 2003, we sent out about 500 questionnaires. Sadly enough, the response rate on postal questionnaires used to be terribly low. We got, I think, about 30 or 40 questionnaires back, less than 10%. But again, that used to be one of the better available tools to get some idea about where species are distributed. Many maps that we know of many species today are still based on hunting records and individual reports by people, either using postal and now, of course, through some of these other tools. In the last, let's say, about 10 to 15 years, just about a decade or so, technological innovations such as camera traps, eDNA, and very easy access to receivers via phones, and also development of apps such as eBird and iNaturalist have added dramatic capability to the conservationists' ability to map species distribution. Not just their ability has improved, their accuracy has seemingly also improved. So that's how things have changed dramatically. I mean, it was, I think someone told us that somewhere in the late 90s, a GPS used to be like a small briefcase. It used to be that big, bigger than our laptops are today. Today, we have GPSs in our smartphones and even watches for that matter. Right. Now, what I'll do is I'm going to lead you to a very small exercise. Now, many of you may follow us on social media. Spot the snow leopard challenge often. I'm not going to share a spot the snow leopard challenge, but I'm going to share a spot the devil challenge. I'll share 6 images, and we will put across a poll along with that. So please answer that poll. Make that selection in that poll accordingly, so that we can get some data from you. I'll just let it go by itself. Those who looked at it carefully in the first go will get extra time. So your detectability might improve, I hope. Okay. I think we're done. Okay, so I'm not going to share the results here. I'll just share the results in another place. Just a second, please. If you look at it here, it's quite fascinating. I'll show you after a couple of slides. Oops. Oh, never mind. Right. I'll show you in a couple of minutes what exactly was the situation, but there were clearly many people who saw the devil when the devil wasn't there. But there were more instances where if the devil was there, some people could not see it. So in other words, what we call, when you see something which is not there, that's a false presence. And when you don't see something which is there, you call it a false absence. Fortunately, we can deal with both of them. It's slightly easier to deal with the false absence than it is to deal with the false presence. And today we'll primarily focus on the false absence. We'll just touch upon how to deal with the false presence, since it requires a little bit of a specific data to be able to address it. Now, so the point we're trying to I'm trying to drive home here is that errors concerning false presence and false absence are the bane of distribution surveys, which have unfortunately been overlooked or glossed over in absence of reliable methods for years. Very quickly, several modeling approaches have been considered in the past, and one of the most popularly used methods used to be that of logistic regression. Now, technically, it makes sense, as this is typically binary data showing either presence or absence. Now, a species can either be present in an area or it can be absent in an area. It can't be in a transient state like, I don't know if any of you have read, but there was a character in Haruki Murakami's Kafka from the Shore called Kafka, who had half a shadow. He had almost a translucent presence. But that's not the case with wildlife. They're either present in an area or they're not. And for them, it becomes fairly reasonable and logical for us to use logistic regression. The problem creeps in when you have to determine species absence. A species could be in plain sight and yet not be seen. If you look very carefully in this image, you will actually notice there is a snow leopard right there. But there's a fair bit of a chance that a snow leopard would have been there and we could have just missed it in plain sight. So if you went out to do surveys, or if people were living in the area and they had never seen a snow leopard, you would have detected an absence for a snow leopard, which is not truly an absence, but it's actually it's technically it's a false absence. It's non-detection, right? Now, this is how our data came through. So 3 people were in Gobi, 3 people did not detect the devil. In Spiti, 6 people did not detect the devil. In Chitral, 15 people did not detect the devil, just because it was such a busy landscape, right? There were so many things happening in that area, you couldn't see it. What's really funny is that some people did detect the devil in Narin. Now, this is a case of false presence. To deal with it, you need certain additional information. We'll come back to that later. But this was fairly reasonable. One person still either pressed the wrong button or claimed to have detected the devil even when it was not there. So the idea being that it depends on how elusive a species is. And on other occasions, it may depend on how skilled the observer is. Basically, it's an extremely difficult task to claim absence with any level of confidence, or it has been very difficult to claim absence. And to deal with this uncertainty over lack of absence data, several methods have been proposed over the last few decades that claim to use presence-only data, be it ENFA, Maxent, Domain, GARP, or several others. Each uses presence-only data to model species distribution. Now, none of these methods are wrong. They all do well as long as you meet the assumptions, the inherent or the underlying assumptions. Uh, if you meet all those assumptions, the models are likely going to produce maps and distributions that are fairly useful. But the concern with these methods is that they do not take into account imperfect detection, which is occasions when the devil is there and you don't see it. In other words, for a species with a great camouflage or ability to remain hidden, you will end up marking its distribution way more sparingly than it actually is. And that's something we did notice in Mongolia, where a couple of years ago, we estimated snow leopard distribution based on direct evidence of presence using um, two different analytical frameworks. And the one that addressed imperfect detection predicted the species in parts of the landscape that none of the other models predicted. And that was quite remarkable. Whereas on the other hand, it also predicted presence in actually in these two areas, it predicted presence where other methods indicated that they could not be there. Good thing is we had the ability and the time and the resources to go and verify these predictions, and camera trapping indicated, it suggested that we were fairly accurate in those predictions. It also identified areas which were being predicted as being used by snow leopards, but in reality they were not. Fortunately, we were able to find out those gaps as well, and that's been a remarkable difference between methods that use detectability and that ignore detectability. So even though some methods claim to use machine learning, what they're really doing is Poisson point process models. There's a bunch of very useful reading material I'll very strongly recommend you to read through. And these are available on as links on the Snow Leopard Network's training workshop on distribution surveys. I think you should be able to use this QR code to access that. The bottom line being that these methods are useful, provided you follow the design recommendations very, very carefully, and you're very clear about what these outputs mean versus what they do not mean. And in my limited understanding, that kind of an advice is fairly sparse, which is why it's really important to read through the limitations and the caveats of the methods before using them. Now, to put it simply, the presence-only methods are extremely sensitive to non-random sampling and also non-consistent detection. Now, just for a change, can any of you spot not a snow leopard, but another rare species called the four-horned antelope in this image? Yeah, basically, this is what the species look like, and it's somewhere right in front. Every species out there has a level of ability, has certain ability to be able to be camouflaged and not be seen easily. So in 2002, Darrell McKenzie, he published what I believe has been a panacea for distribution surveys for its robustness, ease of use, replicability, and ability to address the bane of wildlife studies, which is imperfect detection. Given its statistical robustness and widespread applicability in ecology and beyond, Darrell also published a book that is a must-have if you are interested in using occupancy models for one of its many applications that I'll briefly also talk about a little later. The book is available as a hard copy or as a Kindle copy, which is how I use it most of the times. But it's what I really like about the book is it's written in very simple language with examples, keeping in mind the primary users that are ecologists and not statisticians. And that's what makes it a fairly useful read. Now, coming back to what exactly is occupancy all about, how it works to begin with, and this is something that's extremely important, it starts by discarding any usable information of species detection directly or indirectly, as long as it can be trusted. In occupancy models, the information about lack of detection is treated with the same level of respect, if I may use the word, as information about actual detection. And that is where we come to the next point, which is that of the requirements for conducting occupancy surveys. In other words, what do you need to conduct occupancy surveys efficiently? Uh, there one can classify it into into 5 very basic things. One needs to know how will you sample the presence of a species. You can use camera traps, you can use genetics, you can use eDNA, you can use uh, evidences in terms of pug marks or spoor. People can use even information from those people who are living there and sharing their experiences of sightings. The second important thing is to know what your sampling unit is, because occupancy is a size dependent estimator. For the same distribution of a species, you can have different values of the occupancy estimator depending on how big or small your sampling unit is. It is also a variable that well, in other words, it's something you need to know the variables that affect the probability of a species occurrence, be it environmental variables, be it climatic variables. If there is a hypothesis that you're setting up, you can test its effect on the probability of a species occurring or using a certain habitat. One can use one should try to identify variables that can affect detection of the species. Sometimes the weather could be bad and your detectability could be different. Sometimes the time of the survey could be such that all the animals could be sleeping. So sometimes I mean, sometimes it could be, yeah. So basically, there are various variables which could influence or affect your ability to detect the species, your or the camera traps or the observers. As an example, when we go out asking people for their experience, their recollection of detecting species, uh, some, typically speaking, those who are hunters or those who are forest guards, they usually have a much better recollection than those who might be just farmers or who might just be roaming about or just walking around the area. So sometimes even your profession can make a difference on your ability to detect species. So often these are confused between, but it's fairly important to be able to identify the differences between the two. There could be other variables that could influence local colonizations and extinctions in case of multi-year models. Uh, for instance, mining may have started in a certain area, which may have led to local extinction, or a road could have come into an area, which could have led to local extinction, or a protected area or a community reserve could have come up in an area leading to a species colonization. So these are the 5 key data sets or 5 key things one has to keep in mind to collect occupancy level data. Um, in all of this, the most important piece is that of determining what is your sampling unit. People have in the past proposed it to be to be roughly the same size or bigger than the species home range. Now, while the idea of home range being the same size or bigger than the home range in some cases may work, let's say in case of a frog, which uh, um, uh, you know, which is basically living within the same area. Here is what will happen if you use the same concept with animals that don't have any physical feature as explicit boundaries. Now imagine that's a home range of one of the snow leopards. These are 2 other snow leopards. Fair enough, we're doing good so far. But in reality, the animal ranges will not be so cut tight around the randomly placed grid cells. They will be somewhere like this. So when you estimate occupancy, you might end up overestimating it. You might generate data of presence in these grids. You might end up potentially overestimate the area being used by the species. Uh, in which case, to be honest, a smaller grid might provide a much finer resolution of information than a bigger grid size. I'll come back to the size of the sampling unit in a second, but the other important consideration, which is again, defining the size of the sampling unit is that your sampling effort, whether it's camera trapping or transect surveys, should not just represent a small fraction of it. If you're just representing a small fraction, as you can see in this little transect line drawn there, then you're not, and then you say that whether the species is present or not in this grid cell based on this little transect, then you are stretching your assumptions too far. You're making a huge assumption that if the species was anywhere in this grid cell, you would have somehow noticed in this little fraction of it being surveyed. Ideally, what you need is a representative of the entire sampling unit. It doesn't have to be a straight line, has to cover every nook and corner. As long as it represents the grid cell sufficiently, you will be fairly confident of estimating the probability of it being used or not used by the species. Now, again, I'm coming back to the same point of the size of the sampling unit. While it is not wrong to have large sampling units, given that occupancy is a size-dependent variable, it's most certainly not necessary. The only prime consideration in our limited understanding should be that of you should refer to the objective of the study or monitoring program and the desired biological interpretation of quotes unquote occupancy that the research is trying to encapsulate. Closure of sampling units, which is often considered to be the gospel, it does allow us to estimate if the species of interest was present during the survey. But if it is smaller and if the species is going out and the closure assumption is violated, then we can still answer the other question, which is that of whether the species is using it or not. I'll try to explain it slightly better. Yeah, so if you look at smaller sampling units where a species moves between a few units during the sampling period, it allows us to estimate if the species was using the sampling unit during the survey period, as opposed to the previous case where it tells us whether the species was present in the sampling unit during our survey or not. I imagine if this snow leopard's range was somewhere here, we would have probably estimated its presence in 2 grid cells in a short period of time. And that's where it creates a bit of an issue. Right, so depending on the question you ask, both the probability of a site being occupied and or the probability of a site being used are fairly valid and valuable estimators. The other thing one has to keep in mind is that while we often tend to survey areas that are likely to record detections, obviously nobody wants to do surveys that don't yield, right? If you're going to an area, you would really want to have some detections, whether you're surveying tigers or hornbills or monkeys or whatever. But then, and if you look at other methods which are based on presence-only data, they basically discard these non-detections. So non-detections are discarded, which again is intuitively something that we tend to prioritize. And what it does is it creates it ends up creating a situation where people end up sampling only those areas where you have a greater likelihood of presence. But in case of occupancy, the more random you do the surveys, the better it is. One of the best ways to sample for distribution surveys, as I mentioned, is to go random. Just determine the area that you're interested in and identify a random number of sampling units within this area to be sampled. Now, like this, you see here in this case, in Mongolia, the experts from the country had a very clear idea that snow leopards are only distributed in the western part of the country. They're not living in the steppe. So the moment the moment they identified the area that needs to be surveyed, if they were to survey every grid cell in the area towards the west, it would have taken 3 years or more. what our team did, our team generated a random number of grids within this entire area representing the potential snow leopard habitat, and then they used it to predict snow leopard distribution in the unsampled grids within that. Now moving on, what comes next is the issue of how many surveys are too many and how few surveys are too few. Often when you're starting off with occupancy surveys, you would want to decide how many times or how long do you want the transit, how long a transit you want to walk, or how many times you want to repeat the transits, or how many days you want to set up the camera there for. And typically, to answer this, what we do is we do a pilot survey and use some tools. I'll come to those in a second. But before that, just a quick recollection. In order to estimate probability of detection, what you do need is you need to survey each grid several times, or you need to survey each grid using multiple spatial sampling methods. Now, these 3 trails could be 3 camera traps. These 3 trails could be, instead of 3 trails, you can just have 3 camera traps or 3 genetic sampling transects as well. So the idea being that within a survey grid, when you're traversing it, as you detect a species, you record a presence. As you miss a species, or when you don't detect a species, you just record a 0. And these values of 1s and 0s provide extremely valuable information for the models to be able to predict the probability with which you may detect or miss seeing a species given its presence. In the above case, if you see the first survey, you're surveying the same grid on 3 different occasions. Now, the species are not going locally extinct. That's one of the key assumptions, that in each season or every time you're going out for a survey, the state of the sampling units remains constant. If a species is using it, it will use it during that period of time. If it has stopped use, if it suddenly starts using it somewhere in between, then that's a problem. Then you have to split it into seasons. Or if a sampling unit is empty, it should remain empty during that particular survey of yours. By empty, I don't mean that animals shouldn't come and go. If animal is using it, that means it will come and go. But then in that case, we are going to use it as a probability of site use rather than probability of site occupancy per se. Right. So again, in the first survey, the team went out, they didn't see any animals. The second and third surveys, they saw the animals. So the same grid would generate a data which will be 0, 1, 1. In the case below, it generated a similar data set of 1, 0, 1. Both of them are valid ways of repeating surveys. So, and how many surveys to be done, how many times you need to repeat a survey, how many sampling units you need to survey. To answer that, typically you require some preliminary data, and this preliminary data is generated through a preliminary survey, which could be just about 10 or 15 or 20 sites being visited, and the data collected using the same protocols that would be used later. The preliminary survey provides very coarse but a reasonable idea about what the detection probability and site use probability is likely going to be in that area. It also allows us to modify the sampling protocol. For instance, if you're sampling for too small a distance, our detection probability will be tiny, which in turn will bloat the occupancy estimates inaccurately unless we achieve a very, very high level of precision. So that's where you might either want to increase the length of the transects, or you might want to increase the period for which you're camera trapping to be able to give the animals enough opportunity to be detected. That's the key here. The animals need to be given an opportunity to be detected. If you give them very little opportunity by walking very short transects or doing your camera trapping for very short durations, then you're likely to have lots of zeros in your data, which would mean that you're more prone to inflating your occupancy estimates towards the end. And by the way, I mean all of that, what I'm saying here, there are fantastic tools available out there. Daryl and his co-authors provided splendid means to guide about the recommended number of sampling units and the number of times each of these should be sampled. In very simple words, if a species is sporadically distributed, you have to survey more number of sites, but you can repeat them fewer number of times. You need to do more surveys. Sorry, you need to do more spatial coverage, but less repeat sampling. If a species is very difficult to be detected, then you can do fewer sites, but more number of times. So by doing that, you can do a fairly good job of collecting usable data with minimal cost, field costs. And in a nutshell, again, this is more of a summary out here. To select sites, one has to make sure that they're small enough so that they can be sampled representatively. At the same time, they shouldn't be too big so that you too big so that you are unable to cover them with your sampling. If your sampling unit is also very small, then again, there is a problem because then you will be detecting species which are outside your sampling unit, inside your sampling, within your sampling unit. And that's where it again generates some problems. The other 3 points are fairly straightforward. I can come back. I'm just running out of time, looks like it. And then again, similarly for surveys, one has to make sure that surveys are long enough to provide sufficient detections. And as in case of sites, they need to be able to be, we should be able to characterize them homogeneously by covariates that are of interest. In the same breath, for surveys, we should be able to characterize them homogeneously by environmental, weather-related, or other observer-specific covariates. So that's a very quick run through what occupancy surveys need. Again, I'll repeat, a lot of this information in much greater detail. You may be able to find it in our detailed workshops. But I just wanted to end before ending. I just wanted to run you through some of the usages of this framework. It is a very powerful framework that allows us to answer many questions beyond just where a species is distributed. And I'll finish that, and then I'll hand over to Ian. Like, for instance, within our teams, we've used it to determine changes in space use over time, identifying factors that are likely to influence local colonizations and local extinctions of a species. We managed to use occupancy frameworks on primary surveys using camera traps, genetics, and science surveys by walking on trails in the mountains. We've used it on databases that can bias detectability by providing convenient paths to animals, such as forest roads. But then the good news here is that there are tools that use, in this case, for example, the ones that use Markov chain models to fix that bias. We've also used it on how two species coexist or avoid each other spatially or temporally. And our teams have also used it to correct for people's fading memory and imperfect recollection of encounters. Getting this through reviewers sometimes is a bit of a challenge because often they interpret or misinterpret it as a perception than presence, absence, or detection, non-detection data. Our teams have also been using some of these methods to determine the probability of occurrence of events such as poaching and illegal wildlife trade or conflicts. And soon enough, we should be coming out with some very interesting data sets that determine how the presence of one species influences that of the other. Going forward, we hope to be able to use the occupancy methods to monitor prevalence and spread of diseases using what is called as multi-state models, where probability of detecting something is a process of the methodological and detectability-induced uncertainty. I imagine with climate change, melting glaciers, that have been frozen for millennia, lean in infrastructure, providing access to remote parts of the planet, and illegal wildlife trade proliferating with human displacement and poorly planned infrastructure, disease monitoring, and of course, given COVID, disease monitoring is going to be extremely critical, especially in these habitats, and it will require reliable and replicable methods for the same. So we are hoping to be able to use some of these tools there as well. Right now, despite its strengths and wide spectrum across which it can be used, occupancy methods are restricted in their ability to provide information about or in their ability to not be able to provide information about abundance and density of a species of interest, which is where I'd like to now request Ian to run us through the nitty-gritty of spatial capture-recapture methods. But before I just pass on to Ian, please do take a screenshot of this page or scan these QR codes for the resources that I have extensively used today and earlier in providing these workshops on distribution of species using occupancy framework. Thank you. Ian, over to you.
Okay, so like Koustubh said, I'll be talking about spatial capture-recapture models, which kind of are a complementary tool to occupancy models, in that they go a little bit beyond, they go beyond modeling just distribution, and allow you to estimate how many animals you have in a given survey area. So they have more ambitious objectives, but they also place the kind of data that they need and the requirements that they have are a little bit stricter or stronger than occupancy models. So I'm going to take you through a practical introduction to using SCRs. So it's a hands-on introduction to building these kinds of models in R. But first, I'll just talk generally about spatial capture-recapture models. So spatial capture-recapture models are commonly used to estimate animal abundance and distribution from surveys that they use fixed detectors or detectors at fixed locations to record the presence of animals that are individually identifiable. So in the case of snow leopards, identifiable by their markings. So detection data can come from lots of different instruments: camera traps, hair snares, scats, live capture traps, area searches, acoustic detectors. There's a long list of possible detectors, but the key thing is that you are recording presence of an animal at some fixed location, and you would have a network of these detectors spread across space. So presence can be recorded by an image, a DNA sample, an actual animal if you're doing live trapping, or an audio recording. And spatial capture-recapture models combine two models. One is a model for the spatial distribution of animal activity centers, and I call that a population model. And the second model is a model for the detection process. That's called a detection model. The power of SCR models is that they jointly estimate the parameters of these two processes together. So in the population model, each animal in the survey area is assumed to have what's called an activity center. This is just a conceptual, it's a conceptual tool or trick that represents each animal during the survey by a single point. And that point you can think of as the kind of the center of that animal's activity during the survey. So for animals that nest, you could think of it as sort of like its home center, but that's not necessary. It's just a conceptual center of activity during the survey. And what the population model is a spatial process that quantifies the expected animal activity center density at any point in the survey region. So we estimate a surface that would look something like this picture. where we get a density of animals in some unit of measurement across the whole survey area. And there are two main ways in which you can build a population model. You can either assume that density is constant across your survey area, in which case animals are assumed to come from what's called a homogeneous Poisson point process. It's just some jargon. Or you can model activity-centered density as a function of some covariates that vary in space. But typically with snow leopards, you would model activity-centered density as a function of terrain ruggedness, because the idea being animals prefer certain types of habitats over others. And this is an inhomogeneous Poisson process. And in that case, the intensity varies over space as a function of spatially varying covariates. That's the population model. The detection model quantifies how likely we are to see an animal given how far away we are from its activity center. So we estimate a function that looks something like this, where on the y-axis we have the probability of detecting an animal, and on the x-axis we have the distance that a camera is from an animal's activity center. So a central feature of all spatial capture-recapture models is that you're more likely to see an animal if the detector is close to its activity center than if the activity center is far away from a camera. So the probability of detecting an animal at a detector decreases as the distance from the distance between the animal's activity center and the detector increases. And the detection model is just a function that captures that kind of basic relationship. And there's nothing that tells us exactly what that decreasing function should look like. We just know it should be some decreasing function. So like with many statistical models, making some progress involves two steps. In the first step, we would select some kind of a function or an equation to model the relationship. So this would be an equation with some free parameters. So in the second step, we would estimate the parameters of those functions. So a common function to use is this half normal detection function, which just gives this gives a particular shape to the detection function once you estimate the two free parameters are g0, which is the kind of the intercept on the y-axis, and this sigma parameter, which determines how quickly this function drops off. D is the distance between a detector and activity center. So we would use some kind of an estimation routine to choose the values of g0 and sigma that give us the best fit to our observed data. And that's the detection model. So spatial capture-recapture models need three main kinds of data. They need detector data, they need capture history data, and they need mask data, or data on the mask. I'll talk about these three different kinds of these data inputs that you need to build an SCR model in much more detail in a couple of minutes. So I'm going to be talking about spatial capture-recapture models implemented through this R package SECR, which implements maximum likelihood estimation for lots of different kinds of SCR models. It's a really comprehensive big package that does a lot of stuff, and we'll only really scratch the surface of what it can do. And especially there's usually lots of different ways to do stuff in SECR, and I'll in the hour that we've got, I'll just usually take you through one way to do things, and that's usually the way that I think is easiest, or that I'm just familiar with. So this slide shows two things. The one thing is that you write some computer code to fit an SCR model. If you're familiar with R code, then some of the stuff might make sense. If you aren't, don't worry about it. We'll talk about the different steps involved in the rest of the talk. And then we get some sample output. And these will look something like regression output, where you get estimates and you get some uncertainty about those estimates. Here are the two parameters of our detection function, g0 and sigma. So these are the estimates that we get. And this tells us something about animal density. And again, I'll talk about those things in more detail in the talk to come. So a broad outline for the talk is I'm going to divide the session into two. In the first part, I'll talk about designing SCR surveys with a package called SCR Design, which is kind of an add-on to SCR. So this is basically how you choose where to put your cameras. In the second part of the talk, I'll talk about analyzing the data that you collect from SCR surveys using the SCR package. So before we analyze anything, obviously we need to collect data. And so we need to design a survey in order to do that, in order to collect some data to estimate animal abundance. So this part has three main components. The first is defining the survey region, which is a really important step that's often overlooked. The second step is choosing the camera location, so where to actually go and put up to set up the cameras, and I'll take you through some tools that'll help us to make that decision. And the third step is how to compare different kinds of designs that you might come up with to choose one that's in some sense the best for you. Right, so the first step in any survey design process should be choosing your area of interest, so defining the survey region. So this is the area for which you want to draw inferences. So often for like in the PAWS process, what the focus of interest is in knowing how many animals are present in an area, but there are lots of different other kinds of objectives that one could have. Let's just think about this one for now. We want to know how many animals there are in some area, and we're going to go and set up camera traps in order to find that out. Before we do anything, we need to stop and say, what is the area that we want to know how many animals, we want to know how many animals there are, but in what area? So if obviously the bigger the area we look in, the more animals we're going to find, everything else being equal. So the question of how many animals there doesn't make sense on its own. You have to specify an area to say how many animals are there in this area. And like Koustubh said, with occupancy surveys, you don't need to survey the entire area to know how many animals there are in that area. That's the whole point of statistical modeling, is to be able to take a subset or a subsample and to use, provided that you've done your survey design well, to be able to use the information from that sample to extrapolate to a bigger area. You have to be careful with that, but how widely you extrapolate and things like that. But the point is, that we need to define a region that we want to look at before we do anything. So here I'm going to assume that we've defined a survey region that is enclosed in this red box. This is an area in Tajikistan where we designed helped teams in Tajikistan to design some snow leopard surveys. I won't be using any of the actual camera locations or data from those surveys, but just using this particular area as an example. This is about 3,000 square kilometers. So the kind of area that you'd survey with a single camera trap array of maybe 50 to 60 cameras. So often we've found people go and set up an array without thinking about what the area is for which they want to estimate abundance. And I just want to emphasize that the start of any survey design process should be defining the survey region, which is your area of interest. The second thing is to define the goal of the survey. And there's lots of different possible ones. And I see there was a question in the chat saying, is the most important objective for snow leopard conservation to come up with to estimate population size? And so I think that's a good question. And the answer for me is not necessarily. If you need to, each survey will have its own objectives, and knowing exactly how many animals there are in the region is maybe not, if you have conservation goals in mind, maybe not the crucial factor. But certainly evaluating population size is an important objective in many surveys, and there are many, many clear examples of where to evaluate the effects of conservation interventions, to know whether populations are improving or declining, you need to have some assessment of population size. So the link between conservation and abundance estimates, I think, is clear, or the value for abundance estimates in conservation is clear. But just to highlight that there are many possible goals for a survey, not just knowing how many animals there are. For the PAWS process, that tends to be the main objective that we focus on. But even there are often other objectives that need to be taken into consideration. In a short talk like this, I don't have time to talk about lots of different types of survey goals. So what we're going to do is just assume that the goal is to estimate abundance or density precisely and without bias. So what does that mean? So defining or to estimate abundance without bias, you can think of the end goal of an analysis coming up with some kind of a statement that says we estimate a population density to be something like 1.1 snow leopards per 100 square kilometers. We want that number, that 1.1, to be on average correct. We don't want it to be systematically too high or too low. No, this is kind of a tricky concept because we're taking a sample. So we know we're not sampling the whole area exhaustively. We know that snow leopards, we're not going to see every single one. And so it's not a census. We're estimating a density, and that estimated density is not going to be exactly the true density. We know that. That's all statistical modeling is based on the idea that you have this unknown population parameter that you estimate with a sample statistic. But what we want to avoid is situations where in most of our surveys we're too high, or in most of our surveys we're too low. We on average want to be correct. And the way that what we want is an estimate that's without bias. And the way that we get that is by avoiding sampling good or bad habitats preferentially. So we try and take some kind of a random sample of the landscape. What do we mean by a precise estimate? So a good abundance estimate is never just a single number. You would also give some range, which tells you how uncertain you are about your estimate. So you would say something like, in addition to, we estimated snow leopard density to be 1.1 animals per 100 square kilometers. We estimated the density, the confidence interval for that density to be between 0.9, say, and 1.4 animals per 100 square kilometers. And if we have a precise estimate, we want that interval to be as narrow as possible. The only way to really achieve this is by getting a big sample size, so getting lots of detections and observing lots of animals. That's often particularly difficult for snow leopard surveys, where the population density tends to be quite small, so you don't get loads of animals in any one area, and recaptures are hard to come by. So these are things that are to some extent beyond the control of the researcher. But conceptually, at least, what we want to do is to try and get a confidence interval that's not too wide, particularly one that's useful for if there's some downstream management objectives, that there's not so much uncertainty on our estimate that it's not possible to take some sort of management action. So that's what we're trying to do, define the survey goals. So we can, once we've done that, we can move on to choosing camera locations, and I'll take you through a few different ways of doing this. The traditional way is to set up a grid of cameras. So each of these blue dots is a potential camera location. And here I've got 49 cameras arranged in a 7x7 array. And this is a really good default option, or sort of a first step in designing any kind of spatial capture-recapture survey. When designing snow leopard surveys, it's not practical to give an exact point and say, go and put the camera here, because this point might be inaccessible. It might be somewhere, if we give an exact location to the nearest meter, it might be somewhere where we know there's not going to be snow leopards. And usually, so almost always what we do is to put a grid or an area around these points. This is just a 2 kilometer by 2 kilometer grid. And we say, put the camera in this grid cell, wherever the best possible snow leopard, wherever the location is where you're most likely to see a snow leopard in here. So this could be along a trail, it could be at a scratching post, something like that. So when you design, when you use software to design a camera trap survey, what it will give you is an exact point. But we don't think of these points as exact points. We think of that as some area in the vicinity of the point, and something like within a kilometer of this center of the point. It shouldn't matter exactly how you interpret that, whether it's a kilometer or a kilometer and a half. The basic idea is, if there is an animal whose activity center is in this area, we want to have the biggest possible chance or the best possible chance of capturing it. And that's the basis for setting cameras up along trails or at scratching posts and so on. That's quite different from choosing the best habitat. So notice that we're putting this grid down in some random place in our survey area. We're not saying, well, this is really rugged habitat where we expect to see snow leopards, so we're going to go and put the camera array here and then extrapolate to the whole area. We don't do that. So we choose some random starting point and then put the grid down there. So these regular grid designs with a spacing that's usually determined by the range of the animal's movement. So a conventional guideline is to separate cameras by something like 1 to 2 sigma. A sigma for snow leopards is usually between 4 and 8 kilometers. So that would mean putting cameras somewhere between 4 and 8. Well, if sigma is say 4 kilometers, then it would mean putting cameras somewhere between 4 and 8 kilometers apart. For snow leopards, that means you get way too few recaptures. And so usually what we do is to put cameras something like 2 kilometers apart, with some cameras closer together than this. So we don't often use perfectly regular grid designs for snow leopard surveys, but for they are a really good starting point for spatial capture-recapture surveys. And the reason for that is that they cover quite a large area, but also give you large number of recaptures under this conventional 1 to 2 sigma spacing rule. For snow leopards, it doesn't work so well because the baseline encounter rate is so low for snow leopards that it's really difficult to see the same animal more than once. And so in order to do that, you need to put cameras closer together than the conventional guidelines suggest. Okay, so here's some code for doing this. I'm conscious of time, so I'm going to go through these, just give a sort of a flavor for what the code is doing, rather than go through things line by line. I'll share all of these slides and the code used to generate the design and the analysis parts of the talk after the talk. So in this first, we generate a random starting point, and I do that just by taking a random point inside the survey area. And then I use this make grid command to say I want a 7 by 7 grid spacing 3 kilometers apart, which is wider than what we'd usually use for snow leopard surveys. I specify the type of detector we have, and a count detector just means an animal can be seen more than once on the same camera, can be seen on different cameras within the same session. And I specify this random origin. So these are regular grid designs, and you see it's simple to set up. So there's some alternate new approaches for choosing camera locations that I'll call algorithmic designs. So these are basically designs that try and capture a bigger sample size, or try and place cameras in a way that somehow optimizes some statistical quality criteria associated with the density estimate. So they try and give you more precise density estimates than what you would get from a regular grid design. So what we need to do in order to implement these algorithmic designs in SCR design is to make a grid of all possible camera locations and then let these algorithms choose the best one. There are a couple of different algorithms. So over here, I start with a survey area, and then I generate a grid over the survey area, spaced 2 kilometers apart. And this is my grid of all possible camera locations. I drop ones that are outside the survey area because obviously I don't want to put cameras outside the survey area. So I land up with these 3 lines give me these red dots. And those are my possible camera locations. Often there's some kind of, there's some kind of a filter or a constraint that we know snow leopards won't appear at some of these cameras. So in this particular area, we're confident that we won't get any captures if we put a camera up at an elevation below 2 kilometers or above 4,500 meters. And so I just wanted to highlight this. There's this elevator package which gives you elevation at any point, and then I can filter out any points or any camera locations that are outside of my desired elevation, outside of my desired elevations. So these red points are the ones that are left, and these are all possible camera locations that are between 2,000 and 4,500 meters of elevation. So these are the possible places I could put a camera, and now I let an algorithm choose which ones are best. Before I do that, I'll need this this additional object called a mask. So a mask, I won't have time to get into the details of this, but essentially I said that SECR uses maximum likelihood to estimate the parameters of SCR models. So maximum likelihood is our estimation framework. A maximum likelihood involves some integrations, and doing those integrations over space requires you to basically evaluate what the likelihood is at any point in space. And so we use this mask as it's a grid of points. And at each point, we're going to evaluate the likelihood, or the SCR package needs to evaluate the likelihood. So it'll do that at each of these mask points. And then it'll add up all of those evaluations. And that's basically a way for the computer to do integration. So rather than, an integral is a continuous, requires things to be continuous, but a computer needs things to be discrete or individual points. So a mask is a tool that SCR uses to do these numerical integration calculations. So we need to create one. And all it is really is another grid of points. But I wanted to emphasize that it's separate from the grid of these grid of points on the previous slide, which is the grid of possible camera locations. That grid has a real meaning. Each point is a place where we could put our camera. These mask points, they're just tools that are needed for numerical calculation, or they're just things that are needed for numerical calculations. But we still need to construct it. So we do that by putting an area around our survey area. So I should have said the grid of mask points when we set up a camera. So the grid of mask points should contain all points that animals can come from and still be detected in our survey area. So if we put a camera, for example, right on the edge of our survey area over here, we could capture an animal whose activity center is outside of the survey area. So an animal could come from somewhere over here, travel, and be detected on a camera. So our mask is the set of all points that an animal could have its activity center at and still be detected in our survey area. So we put what's called a buffer around our survey region. And we use that to define the mask. So here I've put a buffer of 20 kilometers around the survey region. So this basically says if an animal is further than 20 kilometers away from the survey region, I don't think that there's any chance of it being detected anywhere in the survey region. So this is usually set to some multiple of sigma, like 4 or 5 or 6 sigma. The bigger the mask, the slower your calculations. And so if that's why we don't set this to, for example, 100 kilometers. It's just going to slow our model down. But if we make this too small, like if we just make this 1 kilometer, that means that animals outside of our mask can be detected at our cameras, and that can bias our estimates. So there's some, I'll talk about that a little bit more later on, but basically there's a decision to be made, and that's how big a buffer do we use? And also how small a spacing between our mask points do we use? I'll talk about some SCR functions that can help with deciding that. But for now, I'm just going to stick in this 20 kilometers and have a mask spacing of 1.5 kilometers. In these two steps, I create this grid of points which are shown in this picture over here. I've just overlain these red points, which are the possible camera locations, on top of the mask. So these are the two things that we need for the algorithmic design operation. We need possible camera location, and we need a mask. Then we need to turn those into SCR objects. SCR needs what's called a trap object. So we use this read traps command to turn our coordinates of possible camera locations into something that SCR recognizes. Again, we just read in this data frame and we say what kind of detector we have. And the same thing for mask. Instead of reading traps, we say read mask. This tells SCR that what we're giving it is a mask. Okay, and then we get to the exciting bit, which is actually creating the design. So we use this GA-optim function. And there are a couple of different algorithmic designs that we can come up with, but I just wanted to show you that the way in which you do this. So we pass in the mask and the coordinates of the possible camera locations, and then we tell the algorithm how many traps we have to use. So I've said we've got 50 cameras to use, and ask the algorithm to tell me where to put them. Importantly, we need to make some preliminary guesses about the likely parameters of our SCR model. So the design that we come up with is going to be optimal, but it's only going to be optimal for these values of G naught or lambda naught, sigma, and expected animal density. If these values change, the optimal design might change. But often for snow leopards, we have a reasonable idea of what sigma and lambda 0 is, and we could find that out by looking at past papers and things like that. But we need to make some guesses before we collect any data about what these parameters might be. Obviously, estimating these parameters is the whole point of the survey design. So you kind of have to get your head around the fact that before the goal is to estimate these parameters. But if we want to design a survey design, we have to make some kind of preliminary or prior guesses about what we think that those might be. In reality, the design shouldn't be that sensitive to these, but you'll need to, it's a good idea to do some sensitivity analysis, plug in different values and see what happens. Then this function has different criterion, and these lead to different kinds of designs. So this design that I'm going to generate now maximizes whichever is smaller, the number of animals you see and the number of recaptures. And that's been shown to reduce the variance of density estimates. So that gives you nice, precise density estimates if you maximize whichever is smaller, the number of animals you see and the number of recaptures. I can set a random seed, and then these are some more technical options which just specify some parts of the SCR model and basically tell the algorithm how long to run for. And I generate a design that looks something like this. So in gray, I've got possible camera locations, and these are the cells that were selected by the algorithm. You see it separates, so it tries to cover as much of space as possible, but it puts these clusters of cameras together to try and get as many recaptures as possible. And that's a feature of this type of design. If I want to generate a different kind of algorithmic design, this design, which uses this criterion equals 6, maximizes the probability of seeing an animal at least twice. And this also gives good precise density estimates. This tends to result in these kinds of designs where you've got a kind of a ring of detectors with some in the middle. So that also gives you, encourages recaptures on different cameras. I see. So there's this final kind of design, but I see I'm not showing this actual design. So this is called a lacework design, which is something between an algorithmic design and a regular grid. I don't have a picture of this because it's disappeared off the slide. But in the slides that I'll post after the talk, you'll see what it looks like. It's basically like a regular grid design where you've got two types of spacing. You've got cameras spaced close together, and then you've got some lines of cameras kind of spaced further apart from one another, and that's called this lacework design. So I specify my survey region, I specify the big and small spacing that I want, and I can rotate these lacework designs by some degree so that they better capture the orientation of say, valleys or ridges in my survey region. Okay, so that's 4 different ways of generating these survey designs. How do we choose which one is the best one? Or we want to do some kind of a comparison. A good way of comparing designs is to look at the expected sample sizes that you might get from each of these designs. So this is kind of a neat thing that you can do. Before you collect any data, you can make some estimates of how many animals you're going to see, how many recaptures you're going to get, and that will give you some idea about how precise your density estimates are going to be. Obviously, this requires quite a lot of guesswork because you haven't collected any data yet. That's the whole point of doing the survey design, but it's good to get a preliminary idea about whether your survey design is going to be up to the job. So whether you're going to collect enough data to be able to fit an SCR model. And you can do this in SCR design with this ENRM function. So I just put in my design, my trap locations, the mask that I've created. So these are things that these are trap locations. This is a mask, and these are some expected parameters from the SCR model. These are the guesses that went into the algorithmic designs. And this says from this design, I expect to get 25 unique animals, 25 recaptures, and 23, these are movements between different detectors. So remember, this design that we created tries to balance this number of animals we see in the number of recaptures. So that's why these numbers are so close together. But if this was a snow leopard survey, this would be a pretty good sample size, 25 animals and 25 recaptures. And we can also use this other function, this is min NR RSC, but this basically gives us the expected coefficient of variation for our survey. So this says from given the parameters that we think snow leopards have and this potential set of camera locations, we should get a CV of approximately 20%, which is pretty good for a snow leopard survey. If this was much bigger, if this was 50% or 60%, we'd know that we needed more cameras or we needed to put them in different locations. So we can compare the 4 designs that we generated, and we can, according to their sample sizes and their expected CVs. And this is just, we don't necessarily want to be too dogmatic and always choose the design that gives us our lowest CV, but it's something to think about, or it's a useful way of comparing designs. So in this case, notice that our grid design gives us a much higher CV. So this is a less precise estimate of density. And this is because we put our cameras, because we put our cameras too far apart. Okay, so that's a set of functions or tools that you can use to generate possible camera locations. It's also good to, what's really essential, to go through a checklist for your survey and see that the design satisfies these basic kind of this basic checklist or desirable properties for any design. So for spatial capture-recapture, it's really important that your array of cameras cover at least one animal home range. So for snow leopards, that's typically something like 300 to 600 square kilometers. So that means you need enough cameras to sample that size area, at least that size area, but also put cameras close enough together so that you get enough recaptures. So typically at least some clusters of cameras that are 2 or 3 kilometers apart. So that means for snow leopard surveys to do both of those things, cover a home range and have close enough spacing that you get recaptures, you typically need quite a lot of cameras. So to do a reasonable survey, but at least for any survey, you should check that your cameras cover at least one home range. SCR models will struggle to fit if you have fewer than 10 animals detected or fewer than 10 recaptures. Sometimes it'll work, but it's risky. So what it means is that the estimation actually fails altogether and you don't get reliable, the model can't fit and you don't get sensible parameter estimates. Sometimes this is beyond your control, like especially the number of animals if you're surveying a very small area. But it's good to try and get these numbers up around 15, ideally quite a lot higher than that. The higher these numbers are, the more precise your density estimates will be. You want to get recaptures at various distances. So this means putting your cameras a variety of distances apart so that when you get recaptures, they're not all at the same spacing. And then you want to avoid sampling only good or only bad habitats. It's only really a problem when people go and only put their cameras up in good habitat, but that will bias estimates. There's a lot more details for all of this stuff at this course that Koustubh mentioned and that I've provided a link to here. Okay, so let's assume we've gone out, put up cameras and collected SCR data. In the 20 minutes or so that I've got left, I'll talk about how to get your data into the format ready for analysis, and then how to do that analysis. So I mentioned we've got these 3 different types of data that we need for spatial capture-recapture. We need detector data, we need capture history data, and we need mask data, and then we can fit our model. I'll talk about these one at a time. So detector data needs to look something like this. So I've put it in the form of a spreadsheet. This is often how I'd get, if I was helping with an analysis, how I'd get given the data once it has been cleaned up a bit. So we've got an ID variable for each camera. So camera A1, camera A2, and so on. Lat long coordinates for each camera. Possibly some indication of effort. So if not all cameras were, if cameras were put up at different times, or if some of them failed for certain amounts of time, then this is sort of the number of days that the camera was on for, or recording for. And then we can have covariates associated with these camera locations. So for example, what was the topography there and what type of camera did we use? So it can come in this in the form of this kind of spreadsheets. Well, this is the easiest way that I find to do it. Set it up in the form of a CSV or Excel file with that kind of format. So then in SCR, we read in the CSV file. The most important thing is we need to convert these lat long coordinates to UTM, because we're going to be working out distances between, we're going to be working out distances. We can't work out distances with lat long. We need to convert to some coordinate reference system where distance calculations make sense. So we do that by converting this spreadsheet into some kind of a spatial data format. So in R, there are these it's SF format, and then doing a coordinate transformation. So this is lat long, and we're changing it into a UTM, and we just need to choose the UTM zone where our survey was done. So this turns things into lat long, and then I'm just going to replace the X and Y, the lat long coordinates, with these UTM coordinates. And for now, I'll only keep the first 4 columns, so trap ID, X, Y, and effort, and I'll show you how to add covariates later. And this is just a little quirk of SCR, that it doesn't read in the covariates if you're reading in from a CSV file. If you're reading in from a text file, it will, but I'm not going to cover that just because often I just think it's a little bit more difficult to do things that way. So now we create this traps object. In the first line, we say where our data, like the name of our data. We say what kind of detector we have. We say if we've got usage data, we set this binary usage to false to tell SCR that we've got a usage column or an effort column, and then we give the name of the trap ID column. So if you remember on that spreadsheet, it was called detector. So I just say where it needs to look to pick up these trap ID variables. And then I can get a summary which I should look at to see whether things make sense. So I've got 49 traps, an average spacing of 3 kilometers. And look at my X and Y coordinates and check that my usage looks sensible. And it's always good to double check that usage is correct. So I can do that with this and just scroll down through the cameras, and you'll see that these 2 cameras weren't on for as long as the other one. So this last camera is only on for 10 days, and this camera was on for 20 days. Otherwise, all the cameras were on for 60 days. So I just want to double check that all of my data, my detector data, is I'm getting what I expect or what I think is right. To add covariates is just you just add them back in this way. So this references columns of my spreadsheet, and this just adds covariates to this traps object that I've created. So now I can use these covariates in my later models if I want to. So that's detector data. Capture history data is another spreadsheet that looks something like this. So you'll need 4 columns, one for sessions. I won't talk about sessions in this talk, but basically if you sample multiple different times of the year, so if you do a sample one year and you do another sample the next year, those would be two separate sessions. Or if you divide up your survey into 2 different areas that are far apart, those can be 2 separate sessions. If you just have a single array and a single survey, that's a single session. So I just put a 1 down, that's what I've got here. And so I just put a 1 down this whole column, but you do need to have a column for session. Then this ID will be an animal ID. So this is animal number 1, animal number 10, and so on. So these are animal IDs. An occasion for camera trap, this will, it's also possible to divide your survey up into different occasions, days or weeks or so on. For count detectors, everything gets collapsed to a single occasion anyway. And for lots of different, for most camera trap surveys, it's appropriate to use a single occasion. So that's what I've done here. We just used a single occasion and a single session. So we just have a 1 all the way down here. And we'd have a detector ID. So this says animal 1 was detected on detector A1 once. It was detected at that same detector again in the 60 days that we were running the survey. Animal 10 was seen on detector B6 and so on. That's our capture history data. If we've got other information on the animal, for example, whether it's a juvenile or its sex, we can add that as additional columns here, but I won't do that here. And again, we go through the similar steps to the detector data. We read in the file, we tell SCR that we've got a captist object, and see here we add in this captist object that we created. It's a little bit confusingly consists of both the capture history, and we also need to tell it where the traps were. So this is the traps object that we created previously. Once we've done that, we can ask for a summary, and we should just check that we get what we think we have. So if we had 57 detections of 22 animals, and we have 49 detectors in this traps object. That's our capture history data. There are lots of different ways to make a mask data. The easiest way is to create it from the traps object. So remember, the mask just contains all those points where animals could have been detected from. So that can be some distance away from the traps, but it shouldn't be too far away from the traps, because we know that if an animal is a long way away from the traps, we have no chance of seeing it at the trap. So the easiest way to create a mask is with this make mask function, which takes the traps as one argument and then puts a buffer around it and some spacing between the mask points. And then the kind of mask that I'm creating is going to be called a trap buffer, which, like it suggests, just is going to put some buffer around and put points inside that buffer. So we're going to land up with something that looks like this. So we put a buffer of 20 kilometers around each of these camera locations, and then add in points spaced 1.5 kilometers apart. So notice here, so I've plotted the mask, that's these gray points. The traps and the survey area is in black. So notice here that I can, from where our cameras are, I can detect animals that have activity centers outside of my survey area. That's totally fine. We'd expect to see that in many surveys. The important thing to remember is that when we look at density and abundance estimates, it's going to be for this mask. So we're going to find out how many animals have their activity centers somewhere in this region, not our survey region. It's an easy step to go from that to finding out how many animals are inside the survey region, but it's going to have to need an extra step. I'll get to that right at the end of this talk, but it's important to remember or to realize that however we set up our mask, firstly, it needs to contain all the points that animals could be detected from. And all of our inferences, all of our conclusions are going to be for that area, for that mask. If we want to extrapolate to some different region, we need to take that extra step ourselves. So, okay, so how do we choose buffer distance and spacing? Well, there's some built-in functions in SCR for doing that. We generate a quick and dirty estimate of sigma using this auto-ini function. And then a reasonable buffer is 4 sigma, so it's 24 kilometers, a little bit bigger than what I've used, but fine. And a reasonable spacing between mask points is roughly like half or 0.6 sigma. So that's a lot bigger than what I've used. So using a spacing less than 3.5 kilometers is totally fine. Remember, more mask points, so bigger buffer and smaller spacing would mean more mask points. More mask points means slower running time, but a better numerical approximation of the likelihood. So it's kind of a trade-off between, you don't want to use too many mask points because you'll wait ages for your models to run. But it's not a problem to use buffers bigger than this or spacing smaller than this. And even the spacing that I've used of 20 kilometers is probably almost certainly fine. It's only if we're using much smaller than this that we're going to run into problems. Suggest buffer is another function that can suggest a reasonable buffer, and you see that this one is much closer to 20 kilometers. So these are just some tools within SCR for helping you to make those decisions. Okay, so if we want to add spatial covariates to the mask, we can add these from raster files or shape files. It's really simple. We just read these into R. We need to project it to the same coordinates reference system that we're using, and then we add them to the mask with this add covariates command. Or if we've got a shapefile, we can do it directly. So add covariates is a useful function. So this is our mask with the covariates terrain ruggedness index plotted. And we can make a final check by plotting the mask, plotting the traps, and plotting the capture history, and just see that things make sense. Okay, so getting into the interesting bit now, that's the data setup. We have to go through that in order to fit our model. We can fit a baseline model in SCR with, well, we can fit our models with this SCR fit function. We need to give SCR fit a few inputs. We give it our capture history. Remember, the capture history already contains our trap location. So that's our capture history data and the trap data. We tell it where our mask is. And then we say what type of model we want to fit. So this is a constant density model. So it assumes density is constant throughout the region, and then no covariates on either parameter of the detection function. So it's basically a fully uniform model. And this specifies this functional form of the detection function that I started out the talk by saying. I gave you an equation. This HN basically uses a half normal detection function, which uses that equation that I gave. So it just specifies what kind of detection function you can use, and there are a whole lot of options within SCR. Okay, so we run the model. That's all you need to fit a model. once you've set up your data. Now we can look at our results. We get coefficients. These are not that useful in themselves, because these are transformed through what are called link functions in order to constrain density. For example, density has to be positive. So we use what's called a log link to ensure that, and that means we need this needs to be back transformed through the inverse log function. And similarly, G0 is a probability, so it has to lie between 0 and 1. So we use what's called a logit link transformation. That needs to be reversed or inverted before we can interpret these things. So a much better way to interpret your results straight away is on this natural scale, which we get with the predict function. This tells you what your link function is, but these estimates have direct interpretations. So our animal density is basically just over, it's 1.5 per 10,000. So this is times 10 to the minus 4. So it's 0.0000014. And that's our animal density per hectare. So to get our animal density per 100 square kilometers, we can multiply this by 100. So this is SCR gives you animal density in units of per hectare, which is not very useful for snow leopards. So usually we turn this into animal density per square kilometer, you multiply by 100, or by 100 square kilometers, you'd multiply by 100 and then by another 100, so by 10,000. So this is 1.4 animals per 100 square kilometers. That's the density estimate from our data. G0 is the probability of observing an animal if you put a camera at its activity center. So it's at a distance of 0. So if you were to put a camera at an animal's activity center, there's still a very small probability of observing observing the animal. This number is per day. If we don't use effort, it's over the whole survey. If we do have an effort variable, like we do, it's per unit of effort. So cameras were on for 60 days. So this is a probability per day. So on any given day, we have about a 4 in 1,000 chance of seeing a snow leopard if we put a camera at its activity center. And this is just because animals aren't very often at their activity centers. And snow leopards are difficult to see. And this sigma gives you some indication about animal movement, and this is around 8 kilometers. So it's 8 times 10 to the 3, which is 8 kilometers. And this tells you that animals will range around about 3 sigma. So the snow leopards in this population range something like 24 kilometers from their activity center. But these two parameters, G0 and sigma, basically just give you a way of knowing what the shape of your detection function is. What else can we do? So most interestingly, we can get abundance estimates with this region N command. So we get estimates of that there are 54 snow leopards. Remember, this is on our mask area, and look at these confidence intervals. They're pretty wide. So somewhere between 30 and 110 animals. Remember, this is on our mask. So I'll talk about estimating abundance in our survey area in a second. Before I do that, I just wanted to mention, if we want to fit a model with spatially varying density, it's very similar, but all we do is we specify the covariate that we want to fit in this syntax over here. So here we say density is a function of terrain ruggedness, and we're using no covariates in our detection function. And we can get estimates. Here we can look at our coefficient directly. And if the confidence interval includes 0, likely this covariate didn't have an impact. So terrain ruggedness for this particular data set didn't have a significant influence, but it almost always does for snow leopard surveys. we can use AIC to choose which model. So we fitted two models here. One had ruggedness as a covariate on density and one didn't. And we can use AIC to choose which one is best. And you can use these AIC weights, which basically are an indication of the strength of the different models. And 82% of the AIC weight is, goes to this null model, which has uniform density. So that supports this idea that terrain ruggedness wasn't actually a significant predictor of density. Okay, so last thing, I mentioned that if we want abundance in the survey region, we need to do an extra step. This is that extra step. So we need to make a new mask that covers the survey region. And the easiest way to do that is just by putting a huge buffer around our traps. So this is a 50-kilometer buffer, which will enclose the whole survey region. And then we crop those points by telling make mask that we've got this polygon that contains our survey area. We'll land up with something like this. So we make this huge mask and then we crop it down to the survey area. And so now we want to estimate abundance for this mask. And we do that by, say, using the same region in command, but we add in this additional argument, which is to say, we've got a new region. It's a mask object, and we pass in this mask. And so this is the abundance estimate that we have for our survey region. So this is the thing that we're really interested in. Just very briefly, the difference between EN and RN. EN is the number of animals that we would expect to see if we could repeat, like if this animal population could be produced many times. So this is an assumption of the SCR model, is that you have the animal population comes from a Poisson, or is generated by this point process. And so if we could take another, like if we came back 10 years from now and did another survey with a fresh animal population, what would we expect to see? So this often is not, this is of more hypothetical interest. What we're interested in is how many animals are there in this particular animal population. And that's this realized abundance, RN. And so this says, for this particular animal population, I think that there are 40 animals. In reality, or for almost all analyses, these numbers are really similar. And even if they don't look that similar just by the eyeball, you'll see that if you look at their confidence intervals, the confidence intervals overlap hugely. So within the kind of uncertainty that we have in our survey, these estimates are actually very close together. So we have around about 40 animals in our survey region. Okay, so that's all for that I wanted to introduce. I know it's been a bit of, this is a kind of a whirlwind introduction to spatial capture recapture, but we wanted to try and provide you with some of the tools and an understanding of the core parts of the SCR model. So I think if I just to emphasize or to summarize in 20 seconds, SCR models build on occupancy models by being able to estimate density and animal abundance for a given survey region. You can do a lot more. You can look at covariate effects on detection or on the population model process. But the core thing is being able to estimate animal abundance. In order to do that, you need to have detection data. So you need to have an array of traps, and your animals need to be individually identifiable. And since this is a snow leopard workshop, I'm going to assume that that's what that's what most people are working on. Animals need to be individually identifiable, and they're recorded in these capture histories. So we need detection data, we need capture history data, and then we need this mask, which is basically just a numerical tool that we can always create that from the traps. Those three things, with some understanding of how these SCR, the R package works, go a long way to for you to be able to kind of implement your own or run your own analyses. But there's loads of help available, which I've got up on this last list. So there's on the GSLEP website, there's lots of resources that Koustubh's spoken about. there's this longer course where we go into all of these issues in more detail. The SCR and SCR design packages have a lot of documentation associated with them that you can find in this website. And there's also a link on this website to an active Google group where people discuss issues that come up. And it's a useful way to get help with your own analyses. And then if you're interested in the algorithmic designs and how they work, this is a paper where we provide some background information on their strengths and weaknesses. I also, very, very happy if you, that's partly part of my role, and well, it's a big part of my role in PAWS is to provide help with survey design or analysis of camera trap surveys, with no expectation of anything in return. So if you're interested in, if you try some of these tools and you find that you want some help, it's just an email away. I'm sure that goes for Koustubh as well, but that's my role in PAWS, and it's really nice to see these tools getting used. So I'm very happy to help, and just an email away. Sorry for going a little bit over 11. I'll draw to a close there and turn back over to Koustubh or Raki. Thanks.
Thank you, Ian. Do we have any questions? Sorry, Koustubh, you're muted. I'm so sorry.
I know I was muted. I just realized. No, thanks so much. I was just going to say the same thing. Thanks, Ian. Some of these, little nuances, they're so valuable when trying to interpret results or even designing. Thanks really for throwing light into those. There's a lot more you can do with spatial capture-recapture and occupancy methods as well. For instance, we're working on trying to understand how dispersal happens between male cubs and female cubs. So how they disperse differently from their mothers. And one example where spatial capture-recapture is coming in extremely valuable, because you can never have that many animals on collar, that many mother cub pairs on collar. So that's one of those many examples where you can extend the usage or the usability of these methods beyond what they're conventionally designed for. Similar with the occupancy as well, I mentioned, we're using it to monitor illegal wildlife trade, for that matter, diseases which are completely out of the box for which these methods were developed. As far as priorities are concerned, I think, Justine, maybe you can jump in, but impacts of climate change and continuing to understand basic ecology of the snow leopards, I think these are two of the key priorities as far as snow leopard research is concerned. Justine, I'm sure there are more which I must be forgetting right now.
Yeah, so I think PAWS is not only for the policy, I mean, it has all this level in terms of bringing all the countries together, right, for a unified method. So it has this collaborative goal as well. But it's more than just knowing how many snow leopards we have globally. As Ian said, it's where they are, because we had to clearly define the distribution, update that distribution where they are. And as PAWS 2.0 develops, we're going to be looking at how to monitor the population over time through key sites related potentially to climate change or other threats. So it is very much linked to threats, which is a priority for many country governments and teams, but we needed a baseline to build on that, right, at the global level, I think.
Thanks, Justine. That's an interesting question. Can we use the same data set to do occupancy and SCR? One requires recaptures, whereas occupancy requires independent detections. Do we need to correct for spatial autocorrelation in occupancy model when using data that were designed for SCR? Actually not. In fact, I'll just answer the occupancy bit, and then I'll let Ian answer the larger question, because this is something he'll be able to explain much better. But as far as I understand, occupancy methods do not get affected by the spatial autocorrelation. They do expect, in fact, there is an expectation that the data will be somehow correlated, the detections will be correlated if a species is there. And so there is that possibility of correlation. There are ways to correct for those correlations, as you yourself mentioned above, using Markov chain models. But apart from that, there's no harm, or there's no clear danger. The only thing is, if you have data where you can run a spatial capture-recapture model, why would you cut down to occupancy? So occupancy is more coarser information, which is valuable for certain things. But if you have data, you already have individual IDs, why would you go for occupancy? You might as well go for spatial capture-recapture and create high resolution maps if you can, than choose the occupancy way. But Ian, over to you.
I think that captures it nicely, Koustubh. I'm not sure exactly of the answer to this. I can imagine some situations in potentially when using SCR data for occupancy might be problematic if SCR surveys are usually done over a much longer period of time, whereas occupancy is meant to be a snapshot. And so if you have animals moving widely, that may need some additional modeling. I'm not sure about that. But I think the main thing is that it does, if you do have spatial capture-recapture, why would you want to? If you have data that's appropriate for spatial capture-recapture, you can get the distribution as well as abundance. And so, unless just for purposes of interest, I'm not sure what the benefits of using an occupancy approach would be.
Yeah, and if you use the same data to do your stratification, then there is a problem. Then you run into a bit of a problem. So I remember there was someone who not here but earlier, who asked that I have some camera trap data, can I use it to create a stratification layer and then use the same data to run capture-recapture? That's if I'm not mistaken, and that's when you get into a bit of a problem.
Yeah, I suppose that might lead to some bias if you depending on how you stratify. Yeah.
There we go. And thanks for your name, Ugyen. Thank you. Yeah, great.
I also wanted to ask Koustubh and Ian, just for awareness for the participants here, and also if anyone asks us in future. I think you guys have just tipped the edge of the iceberg with what you've given us. It's just so much of information.
Yes.
So if people want to know more, are there any potential workshops that they can find out about, or are there any courses that they can look into which will give them a little more of an in-depth understanding?
So Ian, maybe you should go first on this one.
On the last slide, I gave a link to an online workshop, which are just videos covering some of the spatial capture-recapture material. That would be, I guess, the first. If people wanted more in-depth stuff, that would probably be a good place to start. On the materials on the GSLEP website. Those are less. I think are there some videos there as well, Koustubh? I think there are. I think there are also some similar sessions to these. But yeah, I would start with those. Start with the videos. That workshop.
No, I agree, and I think again, but even if you look at those workshops closely, there's still parts and still elements within SCR and occupancy that are not covered. For example, the 4-week session that we did with this distribution modeling, I think it was module 6, Raki, if I'm not mistaken, that also only touches upon some of the applications of occupancy methods. Similarly, the app. I mean, I know for sure that David and others at Cream, they have been organizing periodic workshops, which are week-long workshops, morning to evening, 5 days in a row. You're basically immersed into SCR, and that's, I mean, there's absolutely nothing better than that kind of an exposure. So if there are people who are interested, of course there are costs involved with that, there are travel related issues involved with that, but if there are interests, again, you can write to us and we can write to these experts and explore funding and other issues for those kind of trainings. Similarly, a lot of what I know about occupancy, I learned it through a similar workshop from Daryl McKenzie. He has some amazing training workshops that he customizes the length of it. But again, these workshops are to be paid for. So because Daryl's travel has to be covered, his time has to be covered. So there are that level of detail, if you're interested in, those are possible given resources. But apart from that, most of the other things, I think, in the resources that Ian shared and the resources that are there on the GSLEP website and also our Snow Leopard network, I think they should get everyone a very good head start. And then, of course, people are still doing their PhDs, right, on SCR. So there's so much more that is to be learned and to be done. Yes, Ian's cheat sheets they're the best. They're the best, absolutely.
Thank you, Ian and Koustubh.
Thank you.